It's great to be here. I was reflecting on the fact that I think the last time I was at Maryland was for my pre-tenure tour, which was about, I don't know, 17, 18 years ago. So it's, I think it was last year in like 1999. So, and my research has definitely evolved since that time. So thank you so much for coming. I'm excited to talk about this work. This is relatively new work that I've initiated with two junior colleagues at USC. They are biophysicists, and one of the things that they do, or their specialty is studying particular strains of bacteria. And I've now learned that if you are a bacteriologist, you have certain strains that you learn about, and everybody has their own strain. So I have two different guys and two different strains. So one of the things I want to put forth today to you as a challenge, but also as an exhortation, I think this is a very exciting research area, this merging of the modeling techniques that we as communications, signal processing, and information theorists and control theorists have, and apply it to this new discipline, which definitely does not take the same kind of approach that we typically do in terms of modeling things. So one of the things that I think we can bring to the table is our ability to abstract, optimize, and hopefully prove some elegant statements which give us intuition about how these complex systems work. So in the course of being this editor-in-chief, one of the things you have to do is write editorials and pontificate. And so at one point when I was trying to write my first editorial for the journal, I decided to take a look at where I could find the phrase molecular communication. My thinking being that this notion of putting molecular and communication together was probably a relatively new thing, and who had used this terminology before. And I was humbled by the fact, but not surprised, that people have been actually thinking about these kinds of problems for some time. And I actually found some work from the 60s where this phrase was used. And even going beyond that, in this particular paper, which is essentially the transcription of Francois Jacobs' Nobel laureate lecture. So he was given a Nobel Prize with two other colleagues, and I've written it here because I won't be able to remember it, discoveries concerning genetic control of enzyme and virus synthesis. He actually uses radio frequency communications as an analogy to explain some of the things that they were trying to understand in terms of how viruses occur and bacteria. And so in this discussion of repressors and protein creation, he uses this RF analogy of a wireless transmitter opening and closing doors. He talks about inhibiting signals and how that can create interference. And in fact, in this particular analogy, he talks about these regulatory genes as being the transmitters for this example he's going to give. So I'm clearly not the first person who's thought that there's a connection between signaling and communication and what happens in biological processes. I won't talk about it in too much detail here, but what I will say is if you haven't read Toby Berger's Shannon lecture, the transcription of that, I would encourage you to do that, because there too he revisits this idea. So apparently after Shannon's seminal paper was written, a lot of different disciplines were very excited about the notion of using information theory to try to ask and answer problems within their own disciplines. And people like Shannon, and I think also Fano, were very dismissive of this and thought that IT was only the regime of communications types of individuals and types of problems. And one of the theses that Toby puts forth in this lecture, which I think has tremendous truth to it, is that what is different now for us in this modern time is that our ability to observe and collect data on biological systems has changed dramatically since the 1940s, 50s, and 60s. And so now we have access to these data sets that we just did not have access to before. And so now we have the ability to perhaps do better and more interesting modeling and potentially ask some questions and answer them in this particular space. So what is it that I want to do? I'm going to give you two use cases, two examples of where I think a theory that many of you are very familiar with, Shenner especially, can be used to try to understand bacterial behavior. And I'm going to pick on Shenner a little bit more here to simply say that the postdoc of mine who really opened the door for us making meaningful contributions in this space literally took the insights that he had developed on energy harvesting and networks and applied it to this problem. And I was the one who said, look, this, I think, is the background that you have that makes you right for looking at these kinds of problems. So that means many of you are now capable of jumping into this area. So the two problems I want to look at on the surface are going to seem very different, which is analyzing electron transport in bacterial cables, which are actually filaments, actual multi-hop networks. And on the other side, look at models for quorum sensing. And I'll explain both of these motivations and why we care. Okay, so why bacteria? Why should we care about this? So there are a variety of reasons. They're relatively simple organisms, yet despite their simplicity, they're organisms that we don't perfectly understand. Certainly in terms of being important for processes that we as humans care about, they are everywhere. And in fact, some people actually argue that it was the presence of bacteria, many millions of years ago that changed the environment of the Earth, that actually enabled more complex organisms like you and me to exist. And one thing that's definitely true is that there's much more of them than there are of us. Okay? So they outweigh us with respect to biomass by several orders of magnitude. So they're important for pretty much everything. In fact, I don't know if you've been reading the popular press, but one of the theses that's putting forth now is that a lot of how a lot of our health is determined by the quality of, the bacterial biome that we have within our guts and other things, et cetera. So even though they're relatively simple, as I said, they're not perfectly well understood. So this is the genome for the E. coli. And so I'm simply going to put forth to you that despite their simplicity in terms of being these unicellular organisms, we still have a long ways to go. Okay, so a legitimate question for you to ask is, what's the connection here? Why should I, at least as I'm standing in front of you now as a communications person, be even thinking about these kinds of problems? And what's the right question to ask? And especially in this first part, I actually am going to pose a capacity problem, right? So what I need to do for you in the next 15, 20 minutes is sell you on the idea of what is the legitimate model for which I want to describe capacity and try to compute capacity. Okay? So in this first part, I'm not necessarily thinking about capacity from a communications or signaling point of view. I'm actually going to use capacity as a proxy for something else. Okay? So again, for this first problem, you can ask yourself, well, what does it mean to compute capacity for a collection of bacteria? And this is the actual motivation that led us to consider this particular problem. So this first bug that I care about, Shewanella oenidentis MR1, I now know how to say it, we can call it Shui for short, is of strong interest to a lot of my colleagues at USC, in large part because they are envisioned to be a potential engine for microbial fuel cells. And people are, of course, very interested in this because these are sustainable fuel cells that can be generated. And people are trying to understand how they are going to possibly achieve this. And so what we were hoping to do is to see if we could use these communication theoretic ideas to model these interactions, dynamics, formations of biofilms so that they could potentially figure out how to set the operating points, if you will, of a microbial fuel cell. And so one of the things that we saw, Ken Nielsen isn't my particular collaborator, although he's the one who introduced me to Mo El-Najjar and then subsequently James Bodeker. But he's been studying this particular set of bugs for some time. And this is data from one of his papers from, I think, like the 80s, all right? And so what you see here as a function of time is the amount of current that's being generated per cell, where here per cell I mean per bacteria. And so one of the things that's happening as a function of time is that this biofilm is being created, the number of bacteria in the environment is increasing, but I hope what you see is that at some point around 15 hours, the amount of current that's being generated per bug actually falls off. So there's something happening in the environment there that is dissuading the bacteria from creating more current and passing more electrons. And I will argue, as far as I can tell from my interaction from my colleagues who study this more carefully than I do, they still don't fully understand why this is happening. So in terms of what they know about bacterial interaction, they would have thought that at this point, the cell density was not large enough for these bacteria to be dissuaded from creating this additional current. And so our hope is that, can we, by using our abstracted models, perhaps provide an explanation as to why these kinds of phenomena occur and therefore enable our bacteriologists to set the right operating point. You should be very grateful, because I suspect most of you in this room are theorists. Our research job is much easier than these poor bacteriologists. These grad students have to sit around, they turn knobs, they take video, they literally babysit their experiments for 24 to 36 hours. It's a very time-intensive form of study. And so I think this is one of the many reasons why they're interested in collaborating with us, is that if we can do our abstract optimizations and say, this is the regime that you care about, that will simplify their experiments dramatically, and at least cost a lot less in terms of human time. So I'm going to teach you pretty much all I know about electron transport. It's what's covered in these slides. This is why I rely on experts. So this notion of electron transport is something that has been well understood within a single cell, and it's one of the things that has to happen in order for cells to create essentially their currency for energy, which is ATP. And so you need this flow of electrons through the cell to create a gradient that's then going to drive the synthesis of, as I said, ATP. And so, as I said, this was something that people have been studying for quite a while. They understand it in single cells. One of the things that they don't understand is the following. So I think it's now about 10 odd years ago, in the field, bacteriologists were seeing a phenomenon that they could not understand. So this is a bacterial filament or cable, all right? When you see these white lines, that is literally the end of one bacterium and then the start of another one. And so what colleagues literally would do is they would go scoop sediment, like they would go get dirt from the ocean floor, and then examine what they would find within this dirt. And one of the things that they saw is the existence of these filaments, which was something that apparently was incredibly surprising. And one of the reasons why it's surprising is because the amount of resources at the top of the cable is very different from the amount of resources at the bottom of the cable. But somehow these bacteria are still induced or encouraged to form this filament and create this sort of symbiotic relationship where they each help each other in terms of existing. So bacteria are pretty simple organisms. They only have one goal in life, which is to exist and to duplicate, right? That's about it. And so anything they do is to serve that end, right? And again, I will tell you, people still don't understand why this is happening. Okay, so now I come in and I take a look at this and I say, this is a multi-hop network. I know multi-hop networks. I've studied multi-hop networks, right? We've all probably done some work in this particular space. And so it seemed, at least to me, from a naive point of view, pretty obvious that we would have some tools that might be able to understand why these multi-hop networks exist. Okay, and so this is what we endeavored to do. So I'm going to repeat myself a number of times because there are a couple of messages that I want to leave you with, right? And if you only walk away with one, this is the following, and that is, one of the things that we have to consider in this simple biological system is the fact that energy is not the complete answer, all right? When it comes to a resource constraint, you know, if I design a radio or wireless communication systems, I worry about optimizing my energy and using it efficiently. But there are things related to, let's call it biological happiness, that also need to be taken into consideration. And when we take that into consideration, we get different kinds of results than if we ignore them. So we can't be just engineers when we go to attack these problems. So we're going to call this an information energy coupling. And what we're going to see is that the capacity itself of each one of these bugs to pass electrons is not in itself the limiting factor if I look at the capacity of the overall cable. I'm going to be making a lot of approximations here. So I don't believe that we're at the point where we can actually make strong statements about how these systems work and how they should be designed or optimized. But you know, each paper we write, we learn something new. And what I'll also tell you is that the work I'm going to talk to you about today, pretty much after it was published, my biophysics colleagues realized that our model was not correct because further experimentation and observation had been done. And so they realized one of the things that they thought was happening was not happening. So again, as a theorist, this is exciting because if the model is wrong, you get a new model and then you get new results, right? So this is going to be a fruitful area for some time. So I don't want to imply in any way that our biological colleagues don't do modeling. They definitely do. They just take a very different tactic to study these large-scale systems relative to what many of us might do, which is to say that they are very careful. They look at the master equations for chemical diffusion and all these different processes. They collect a very large mass of ODEs and PDEs, and they toss it into some kind of numerical solver. So anytime you want to change something, you turn a bunch of knobs, you have to recompute everything. And so it's enormous complexity. Now, what we're trying to do is to simplify things, and in discussion with our biological colleagues, figure out what is sort of the minimal thing that we need to model in order to get some understanding and still have it be a model that they will take seriously. So what we want to do is take advantage of stochastic queuing models, which we're used to using in wireless communications. And this is the translation that we're going to do. One of the things that we are worried about is measuring and analyzing what's happening to both chemical and electron concentrations. So I want you to think of molecules or electrons as quanta, like packets, all right? They're going to be fed into queues. These queues are going to be interconnected in ways that are not typical for us from the wireless point of view. So there are certain constraints and equilibria that need to be maintained. We know that there's variations in the environment and what's going on per bug. We're going to capture this stochastically, as we often do. And the result is actually going to be a Markov decision process model that we can now analyze. Now, for what I want to do, my ultimate models are actually going to be very complex.
Is the underlying model the Q, the Markov chain underlying the Q?
Yeah.
What is the Q representing?
It literally represents molecular concentrations or electron concentrations.
Okay.
Okay? So I have rates, I have arrivals, I have all of the things that we're used to dealing with in wireless networks. So another piece of this puzzle, which I'll allude to at the end of the talk, that we're also working on from a parallel direction, is that we still need other tools, I believe, to really get the complexity of this system down, all right? So you're going to see me propose something that has complexity associated with it. So I don't want to imply that somehow, we're that much better in this complexity question, but I think we're getting ourselves to a point where we can do some optimization.
So is the connection between the past and the current fluid flow approximation, or is more?
I'm sorry, one more time, I just missed.
The connection between the now and the before fluid flow approximation, or is more? Take Q's, go to fluid flow, get ODEs, get PDEs.
Yeah, exactly. These things are all connected, right? So even the fact that we're using Poisson arrival processes, I mean, these are all, you can see these as discrete approximations, right, of some of these continuous time processes,
okay.
All right? So this is it. This is the model, all right? So I have previous bug, current bug, leading to next bug. Let me walk you through the 4 queues that I need in order to describe what's going on. So my first queue is literally electron transfer from the previous cell. Okay, so as I said, I'm in this cable, I'm getting electrons from the previous bug. The other thing that's also happening is due to molecular and chemical processes, I have another queue here where things diffuse in from the environment, okay? And so this also creates a flow of electrons due to this electrochemical energy, right? So this is my second queue here, which we call the NADH pool. Then these two feed into 2 other pools. One is the construction of ATP, and remember I told you this is the cell's energy currency. And one of the things that is different about bacteria, say, at least me, is bacteria don't overeat, okay? And I know this seems like kind of a joke, but this is actually part of why this problem is exciting and interesting, because this is going to create a state-dependent environment, right? Where my input is going to govern the behavior of my system, right? Again, wireless communications, Mother Nature doesn't care if I'm sending, 50 kilobits per second or I'm using a different carrier frequency within a certain regime. The additive white Gaussian noise is still the additive white Gaussian noise, right? Here, however, the processing of electrons is a function of the rate of electrons that are put into the system. And so there are 2 things that I have to worry about. If this queue gets too full, the bacteria says, whoa, back off. I mean, you can send me the electrons, but I'm not going to process them. And if it gets too empty, the bug dies, all right? And so these are the 2 extrema that I have to worry about. So I need to be in a happy zone, in between these. One of the things that we need to work on that we have not achieved yet is the appropriate model for how the bacteria backs off in terms of processing electrons as a function of the input rate. So we picked something very arbitrary right now, but it does provide some interesting results. So I have electrons going into here, and then I also have electrons going out, right? So when they go out in two ways, they diffuse into the environment, and they also get transferred to the next bug, all right? So you can see I have these interconnections. There's some of these extreme points I need to worry about. So at this point, this is essentially what we have. And now we're in a regime where we can actually do some computation, do some simulation, and see how things are going. And so I'm going to go back and forth between these two extreme, right? Where if ATP goes too low, the cell's going to die. If ATP gets too high, then I have this so-called clogging, right? And so, as I said, the bacteria is not encouraged to push as many electrons through the system as possible because that is not behaviorally good for them. So one of the things that we wanted to try to do, and this is again another one of those moments where I'm grateful for being a theorist versus a bacteriologist experimentalist. We've started working with these colleagues many years ago. We're in 2016 now. I mean, I think it's been 4 or 5 years at this point. First, going back and forth on modeling, you think you've understood someone, then you realize that you haven't. So we had hoped that we would collect data on Hsieh that would give us information about the NADH pools and the ATP pools. And it turns out that this is incredibly challenging to do. So we can get ATP levels, but apparently NADH is very hard for this particular bug. What I do know is that when that paper gets written that explains how they collected the data on both ATP and NADH, I will have the least important authorship in a Nature paper, okay? But what I'm excited about is that I think we've pushed them to think about how to do these measurements, and they perhaps would not have thought about these measurements before. So we create the models that are associated with those cues. We essentially solve an expectation maximization algorithm to get parameter values. And so what we did do is take data out of this particular paper in order to understand whether our models were reasonable or not. So why is it easy to get NADH and ATP levels in yeast? And part of it has to do with the fact that the, what do you want to call it? The resource there is glucose, which is a very large molecule, has been very well studied, and it's much easier to do these kinds of experiments. So this is experimental data that we've put on, so obviously we're taking the experimental data, we're using that to drive our estimation algorithm, which is going to give us the parameters, and then we're generating what we think the ATP level should be as a function of time if there's an impulse of glucose added at this time. Yes, Prakash?
Previous slide.
Yes.
You have that product form which signifies some form of independence. What is it?
Yes, so we are making assumptions about how states evolve. We're assuming that things are Markovian, et cetera. So it's sort of the simple general models that you would think of. So I have a picture later on, but from that series of interconnected cues, we can come up with the Markov chain. It's actually fairly regularly connected, and we use that to drive all of the estimation here.
And campaign is the number of pieces, the number of systems that you have?
K here.
N.
Oh, capital N. I'm embarrassed to say that it's escaping me right now. I want to say actually that it's a function of the number of observations that we're taking.
But in your picture, that filament, how many pieces did you have?
Oh, okay. So, yes. Hold that thought for just a second. Okay.
I'm not moving.
Yeah, good. So now that you haven't moved, our experimental data is actually for a single bug, right?
But going back to your model, how many pieces do you have?
So they're typically based on the measurements, it would be about 1,000.
1,000, that's 1,000 change.
Yeah.
So the conditions don't change from bottom to up?
No, they do. So and that's part of one of the things that needs to be introduced. So
It's captured how?
it's captured in a function of the diffusion rates of the chemicals into the various
So the
cues.
lambdas will be dependent on
The lambdas will be
requirement.
dependent as a function of time. And I'll be honest with you, we're not there yet, right? Because if I want to handle 1,000 of these guys, my overall Markov chain is going to be enormous, right? I mean, I probably even can't count that high. And so this is why, from a completely different angle, we've been looking at sparse approximation strategies to figure out how to compress these graphs and then do optimization on compressed graphs.
Because they connect this way, there are no jumps in the connection. Right?
There's no jumps,
So
as far as we can tell.
You can have 1,000, 100,000, but they will be very sparse, the connection.
It will be very sparse, exactly. So not only will the graph be sparse, the behavior, at least what we've seen for wireless networks when we do a similar kind of thing, is that even though the state space is very large, you end up living on a pretty low dimensional manifold. But I will say that graph compression is more challenging than we thought it was going to be. So this is still something that we are messing about with. Okay, so here's my single bug. I've gotten rid of the 2 Qs, right? The whole motivating factor here. But one of the things that we see is that we have very good experimental fit. And one of the things I can tell you is that when we first showed these results to our biophysics colleagues, they said, this can't be right. There's no way that you can get a model that fits things this well. So, and so we redid everything. We walked them through all the code and everything that we were doing, and we convinced them that we were doing the right thing. So as I said, we're still waiting on data for Schumanella where we hope we can, again, see whether these models make sense given the bug that we started out with. All right, so now I'm going to get even more abstract and even more idealized, because one of the things that we really wanted to do was to try to study this cable, as we talked about. And I wanted to be able to see if we could assess the maximum amount of electrons that we could push through this system. So I'm going to go through a lot of idealizations now, but I will say the resulting models and the results that we have are, at least to me, very interesting from a theoretical point of view. Okay? So I'm now going to ask you to withhold even more belief and say, okay, now I want you to think of this collection of bugs as a channel, all right? And I'm going to use capacity as a proxy for assessing how many electrons I can push through, which is then going to give me some sense of how much current a cable can generate. Okay, and again, this is one of these problems where you have to take an incredibly long-term view, right? Because ultimately, I want to hit biofilms, which is a complex, randomly connected set of bacteria for which there's going to be also a relatively complex graph that I need to analyze. But I don't know, I'm optimistic. As I said, the problems that get revealed are just so interesting, and they ask us to think about these problems in a very different way. So, all right, I'm going to tell you things that many of you are intimate with, which is to say, all right, people know what the capacity of a multi-hop link is, right? People have been studying this for years. And you can show via cut-set bounds that it's essentially the capacity of the worst-case link. Right? And you can show that decode and forward can achieve this. Well, of course, the challenge here is that our conventional sort of doubly thinking about this is that each of these nodes are sort of independent systems, and they operate relatively independently. And this notion of my link capacity being a function of the flow in, that's not something that we've worried about, right? So we're now in this regime where we have a very interesting communication over channels with state problem, right? And so this is what we're going to attack now. And we have this coupling between the links, all right? And so this is now, again, like I said, the big abstraction that we're going to do, which is that I'm going to think of my input as an encoder, and I have a decoder on the other side. I'm going to aggregate all of the bugs together to be my channel. So my cable is contained everything in here. And I'm going to look at this notion of the cable state, which is literally going to be the number of electrons in the system. Okay? And as I said, this is going to affect my ability to transmit electrons through the state. So I have this coupling between the energetic state of the cells. And I go back to, to this tension that I tried to sell you on earlier, which is energy dimension versus information dimension. So I'm going to do even more simplification, all right? So now what I'm going to say is I'm going to recognize that the number of electrons in the cable are going to affect my communication, as it were. And so I'm going to introduce this so-called clogging function. And I told you this before that we picked this arbitrary linear function here. I don't even think we're in a position yet to take measurements where we could even see what the right model is. But once I have the model, I can plug it into my computations here, and I can get the appropriate answers, right? So if the state is 0, then that's actually bad because technically the bug would die, but we've had discussions, these existential discussions about does the bug really die or what is death? But so we're going to assume that And we will see that the bug rarely does this, but if there's nothing in there, I send everything through. And as the number of electrons S increases, then I'm going to tamp down on the processing I can do. Okay? So A of S is the ratio of electrons that I'm going to accept through the cable. Okay? So now, this is my simplified system. I am going to assume Poisson arrivals. And one of the things that you can, one of the things that we show, which is not too surprising if you look at some of the prior work that especially Weiner did on related types of problems, is that, so what is it that I'm trying to design here? I'm trying to design this rate lambda of t, right? So this is essentially my information-bearing signal, which is going to govern the rate of those Poisson arrivals through the system. And I'm going to limit it to be between, you know, some minimum and maximum value. And so my input now is going to be this x, which is going to be this Poisson process where I have the rate that I'm designing, but also it has this scaling factor, right, which is going to tamp down on my ability to process. So in a way, I've taken the clogging function of the cable and I've moved it into the model of my input. Okay? So if S of t of large, A of S of t is going to be quite small, and that's going to affect this rate. Okay? And then I'm going to have this output flow here, and that's what I want to assess. So there's a lot of interesting work in this particular space. Again, from a wireless communications point of perspective, as a function of whether you are controlling things or not controlling things, do you have perfect channel state information or not? We're going to assume that we do have perfect CSI. But one of the things that we're actually going to show is that feedback is not necessary in order to achieve capacity of this system. We are going to ride heavily on this prior work by Chen and Berger, which is interesting. When they did this in 2005, I don't think it was for a biological problem, but you see subsequently that Toby uses this paper and its results to come up with some answers and some of the biological problems that he's been studying. So we have a Markov state controlled by our signal, and we're going to assume that we have full CSI. We're going to adopt some of the modeling assumptions that Weiner has in his 88 work, where he looks at static Poisson channels. And I want to mention this work by Stefan Moser during his PhD work. They look at upper and lower bounds for certain systems, but they don't give us much intuition in terms of operation, right? And they're challenging to parse. Okay, so what's the first set of assumptions we're going to make? Oh, I'm going too slow here. So I have an assumption of a small slot duration, and I'm essentially going to assume that the probability that I have more than one electron either leaving or coming in is essentially 0. So, you know, this is 1 minus electron, this is 1 plus an electron, so this is the chain that's considered here. I'm only going to consider these adjacent states. And so, again, we keep simplifying things more and more. One of the things that you can show is that binary inputs are optimum. That's one of the results that we have. Again, so this is my simplified problem here. So in the Chen and Berger paper, they show that you can write the computation of capacity as a dynamic program. And we can do that as well in our particular case. And so essentially, the reward here is the sort of mutual information per state, which is conditioned on this past state S as well as our control function. One of the things that we can show, as I alluded to earlier, is that capacity is achieved by feedback-independent inputs, even though we have this Markovian system. And there are other things that we can also show that simplify computation dramatically. So one of the things that you see is that the important thing that you need to compute is the average intensity, all right, versus knowing what the intensity function is exactly. And so this resulting stationary distribution is then only a function of this mean intensity, which means that we can now optimize encoding functions over the mean intensity only. So that simplifies things a lot. And as I said, we can also show that the binary signal is optimal. Honestly, this result is relatively intuitive once you see the Weiner paper. They have a similar result in there, even though they look at static systems and we have this dynamic system. Okay, so I in no way want to imply to you that bacteria are myopic, all right? But of course, one of the things that we do when we look at these dynamic programming approaches is because of their computational complexity, it's quite common to look at a limited horizon, see what kind of tractable solution you can get, and then typically you say, okay, in many of the cases that we see in our engineering problems, the myopic solution often does not cost you that much in terms of a loss of performance relative to the overall optimization, right? This is not something we're actually going to see here. So one of the things that is provable is that the mean rate for the myopic case is larger than the mean rate for our optimized system. So the implication for us here is that we already know that in the myopic system, you're going to try to push more electrons through the cable than the optimal case. So what you expect to see is that this is going to cost you in terms of your capacity because you're going to get into an environment where the clogging becomes more active than in the optimal solution. And so, okay, I've already given you the preview here. So if you look at the achievable rate here, you see that the optimal system, which takes this clogging into consideration because you're looking at many steps ahead, gives you a higher rate than the myopic system here. So what the myopic system is doing, it's sacrificing the happiness and the health of the bug in an attempt to push more electrons through the system. And of course the bug says no, or the bugs collectively say no, we're not going to overeat. Okay, so in the last 15 minutes, I'm going to quickly show you another problem. Yes?
I cannot model this as a partially observed Markov chain problem.
No, you definitely can. You definitely can.
We can.
Yes.
And then you're going to get the, so the question is, do you get an application of separation theorem here or not?
I don't.
In that case, the optimal control is going to be a function of the conditional density of the state given the observations.
Right.
And it will not necessarily be the mean, but it may be a PMF.
So I don't have enough intuition as to how this clogging function would affect the results in this case.
But in that setting, the fact that the myopic allows you more, it's not unexpected because it doesn't take in the myopic, it won't take
no.
memory that I'm going to hold.
No. I mean, the fact that we get these results is,
Because you have the clogging mechanism, you're going to call it back.
Absolutely. So the fact that we get these results are not surprising at all. But it's still nice to be able to confirm them, rigorously that our intuition is correct.
Yeah, let's be the other one.
Okay, so the next application is going to seem very different. But in a sense, these cues also are very useful because, again, we're passing quanta around, okay? So does anyone know what quorum sensing is? Okay, well, besides
There's a whole group here.
So let's talk about this particular application. So this is a picture that I stole from this particular web page. So essentially, bacteria create compounds that they release into the environment. They also have the capability of sensing these compounds. And the idea is that there are certain kinds of behavior you don't want to engage in unless there's enough of you, hence a quorum, all right? And so you have these three parts of compound synthesis, recognition, and then once you've exceeded a threshold, a response. And what's interesting, and this goes back to, and I'll show you a slide of this earlier, is that one of the first people to actually analyze this notion of quorum sensing in bacteria is this colleague of mine, Ken Nielsen. And so this is actually the original paper. He's often considered to be the grandfather of quorum sensing. And there was essentially this form of squid that would bioluminesce under certain conditions. And people thought it was the squid that was luminescing, but it was actually the bacteria that lived on the squid that formed a quorum and then bioluminesced. And so this is what started a lot of this. If I can go back here just for a second, one of the reasons that my colleague James Bodeker is interested in quorum sensing is that there are certain bacteria that when they achieve a quorum, an infection occurs, right? And so the idea here is, can we suppress certain kinds of infections by preventing them from starting at the beginning without resorting to antibiotics? So the idea is not to kill the bacteria, just to get them to play nice, if you will. And so if this can be done, this has huge implications, well, in terms of many things, but pharmaceuticals for sure. So intriguingly enough, in this particular case, our objective is to suppress quorum sensing, all right? And we want to see if we can engineer a system to do that. Okay, so there are a lot of pieces here. And this is also a place where interacting with these biophysics colleagues gave us a view and a perspective on the right model that is fundamentally different and certainly much more complex than what we've seen when we look at other people like myself that are playing around with this. So a lot of people simply say, okay, I'm going to measure a compound, I'll create a model for how that compound is created, and then when it exceeds a threshold, I'll model things that way, right? Which is a relatively simple model. This is the simplest model that James would allow us to study, right? Because he felt it contained the minimal number of interactions and processes. So I hope you are good pattern recognizers, because you will need it a little bit to track what I'm going to do. So that compound that gets created, that's called the autoinducer, all right? And that's something that we want to measure. So it's the level of this autoinducer that's going to decide whether the population forms a quorum or not. We have these things called receptors, and after an autoinducer binds to a receptor, it creates a complex. And this complex, in turn, is important for the creation of other things. I also have this notion of the production of autoinducers, which is done by these things called synthases. And then one of the things that's important is for these complexes to bind at specific sites on DNA, which then in turn result in all these other things being created. So one of the other things that's going to be created is the bad stuff, which is the virulence factor, okay? So you now, in the next 30 seconds, you will know as much about gene expression as I do, but this is the relevant stuff. So you have this genomic DNA. It has these binding sites here, which are shown by these orange, red, and blue boxes. And those complexes, all right, bind to these various sites, okay? And as a function of these bindings, other things happen. So as I'm talking about this, I'm sure a lot of you are thinking, oh, there's the probability that this is going to happen, there's a probability that's going to happen, and that's exactly how we're going to model things. So we've gone through this again, but this includes some of the assumptions that we're going to have. So now, these complexes are going to bind to these various sites, and then each one of these three things can possibly happen. I can create more synthases, which in turn creates more autoinducers. I can create receptors, but I can also create these virulence factors. All of these things can degrade. We're going to assume that cells can duplicate, but we're not going to worry about them dying, okay? Which is something you could add into the model, but it's just another layer of complexity. So now here...
So which one of those you produce depends on the binding site.
Exactly. And
every one has an index.
Yes. So each one of these sites has a different probability of binding. All right? And so here, this is the notation you want to keep track of. Autoinducers is the only global variable. It's the entire environment, right? How many of these compounds do I have, right? Then I have per cell, so you can quickly see where the complexity is going to get kind of crazy. I have receptors. So each cell has its own number of receptors, each cell has its own number of synthases, and each cell has its own number of complexes, each of which can bind to one of these three different sites. Okay, so here's the queue now. Okay, so now within each cell, I have a queue for each one of those things, right, as I'm creating them. And then I have the global queue, which tells me the number of autoinducers in the entire system. So at this point, things look really bad, because if I do more than one bug, suddenly I'm keeping track of a ton of different variables. But the good news is that you can actually show that the aggregate values here, the number of cells total, and the total number of complexes, the total number of receptors, and the total number of synthases, this is actually a sufficient statistic which is the only thing I need to track in order to assess whether quorum sensing is going to occur or not, okay? So this is very good news, because now I only need to track aggregates, I don't need to track individuals. So let me show you a couple of simulations and then show you some data that we were able to get from James, which essentially says that there's more that we have to do. So for reasons that are still not completely clear to me in terms of the interactions of these systems, cell density in an open system tends to be higher than one in a closed system. So in a closed system, nobody can go anywhere, and in an open system, things can leak out. This higher cell density leads to a larger larger value of autoinducers, and as a result, quorum sensing is invoked sooner in the open system than in the closed system. So what do we see here? Here we see the cell density as a function of time. Open is blue, red is closed, And you can see that in this closed system, the cell density, is slowly increasing here, but it's much lower than the blue lines here. These vertical lines correspond to the point where quorum sensing is activated because the number of autoinducers exceeds a threshold. All right, and then you see this here too with the autoinducer concentration. Again, in the open system, which is blue, it's growing faster than in the red one here. Okay, so this is pure simulation, pure synthetic. We're tracking aggregates, et cetera, et cetera. Let me now show you some experimental results, all right? And so one of the things that's interesting here is that if we look at the cell density in the closed system, we actually, again, expectation maximization algorithm to set parameters, and then we plug everything in, we get a pretty good fit in terms of predicting the number of cells within the environment. And this is data that we took from James's lab. We had no hand in how the data was collected. I don't think he even knew that we were going to be using it. We were actually, so one thing that I've learned between my two collaborations, collaboration 1, we said we would love to have these experiments, and we're still waiting. Collaboration number 2, what data do you already have, right? So, and how can we design our models so that we can adapt the data that you've already collected? So I've learned my lesson, that experimentation is hard and I should rely on what exists rather than being hopeful for what they can collect. Okay, so the closed system data looks really good. If I go okay, and this is also looking at synthase concentrations, and there too, we get a decent match. But one of the things that I want you to take a look at, and this is a little hard to parse here, but these boxes over here correspond to, let me make sure I get this right, experimental data, and this is actually for a closed system, and in this particular case, our models are not actually as good. And so what we see is that quorum sensing is induced sooner than would be predicted by what we have. So the key thing that you want to see here is that the red and the black don't jive very well.
Is it
Okay.
both for closed systems?
This is both for closed systems, yes.
And for which one of the bacteria?
This is, I'm assuming it's Pseudomonas aeruginosa,
Okay.
because that's what James tends to study.
Missing a mechanism?
I think we are, but we need to understand this further. So literally what this is the result of, hey, I have a set of machinery, I used it well on this other system, can I quickly come up with something that makes sense in order to handle this? One of the challenges here is that there are some functions that are embedded in our models which I don't think are very well justified by our physics colleagues, but they tend to throw them in because it makes the data work. And so we've been trying to work with them to understand, well, what do you actually think is happening, and then can I design something that makes sense? So we're not there yet, but this is very early days. We have this results published, but it's in a position paper. It's not what I would call, a theory or analysis paper here. So, okay, so there's more bad news, if you will, which is, the model that I gave you was the simplest one that this biophysics colleague was willing to have. This is actually the signaling and interaction system for Pseudomonas aeruginosa. And what you see here is that there are many more molecules involved. There's much more interaction. They have different kinds of processes going on, some which, are stimulatory, some that are suppressive. And so I think, this is much more complicated than we've started out with, and the question is, what is the right abstraction? And I'm not quite sure yet. So, this is essentially what we've done, right? We've taken these sort of queuing models, we've built Markov chains that seem to make sense.
When you said in your theorem that the sufficient statistic is the aggregate, I'm surprised that the topology and the location of the cells doesn't play any role.
I'm sure it does, but it's not something that we've taken into consideration.
That's what is missed in the...
Very possibly.
Because if you just look at the aggregate...
Yeah, no, absolutely. And so you're absolutely right. That is...
they have to communicate chemically
yep.
to say, to see the
absolutely. No, I agree 100%. Baby steps.
I agree. I've been there.
So as I said, we've gone through this process in general where we've looked at these queues to capture the inflow and outflow of these various quanta. We've built these Markov chain models. And as I told you, quickly the complexity of this is going to get very high. So let me just end things by saying, in this parallel effort, we're trying to use a different set of strategies to allow us to compress graphs. It's based on using compressed sensing and sparse approximation ideas that were developed for time series analysis that are now being considered in the context of so-called graph signal processing. So we have a collaboration with Antonio Ortega on this, and at least for one of the initial works that I did with a former postdoc, Marco Lavorato, who's now at Irvine, and Andrea Goldsmith, is that for learning, so in this particular work, we had a graph describing a wireless communications network. Each node in the graph describes the behavior of the entire network simultaneously, right? So how many packets are in the system? How big is this queue? Am I sending ACNACs, all that kind of thing. And what we were trying to measure is that as a function of that particular node in the graph, what is the throughput associated with that? So that's the graph signal that I'm trying to learn. And one of the things that you could see is that, with these sparse approximation methods, you can reconstruct that graph signal incredibly well and much faster than traditional Q-learning types of techniques, where you essentially have to visit each node of the graph multiple times to essentially approximate the probability transition matrix. And so this is supposed to give you hope that these graph compression techniques might have some utility. But again, there's much under the rug here. This is a centralized system. It's not distributed. I mean, there are many, many things that need to be done. All right, so you've been incredibly patient. Thank you. So in summary, these, I think, relatively simple queuing models, I think, actually have a place in analyzing some of these biological systems. We still have a ways to go with respect to complexity. One of the things that I very much like about this problem is this notion of communication over channels with state. I think this is a research area that has not been heavily pulsed, if you will, in the IT community. It was a while ago, but I see now a great opportunity for us to revisit these problems that make complete sense for this particular application. And this quorum sensing modeling, I think, there's much more that we need to do. I don't think the model is right yet. So these are some of the things that we've been working on. I want to thank my collaborators here. So James and Mohammed, or Mo, are the two biophysics colleagues I talked about. Sahand is a former PhD of Mo's. Nicolo is this former postdoc who's now an assistant professor at Purdue. And let me end with, self-promotion, well, not really self-promotion, journal promotion, which is we're looking for papers for the new journal. I'm inviting people to write things to help jumpstart things. I'm very excited about what this journal can be. One of the things that we've seen when we interact with people who do a lot of research in biological systems is, and I hope I don't offend anyone here, but biologists don't care about theorems. At least that's been my observation, or even potentially algorithm development. So I believe that we can be a home for where this sort of systems theoretic approach to biological system analysis can occur. So I will take your theorems, I will take your approximations and your idealizations, and your algorithm development. Okay? Thank you very much.
Thank you.